Greetings and welcome. And thank you so much for joining us today for our second Best Practice Webinar Series event. I'm Allison O'Connell, and I'm Head of Marketing at Highwire, and I am your host. In one moment, I will hand it over to Highwire's founding director, John Sack. But before I do, I have housekeeping items to share. First, this event is being recorded with the intention of sharing with the community. We will share it immediately following the session, and our full event will be about 2 hours. The first hour is dedicated to presentations, and the second hour will be dedicated to panel discussion. Questions from you, the audience, can be asked at any time in the Q&A box, please. And we will do our best to fit in as many of those questions into the panel discussion as we can. Following this session, we will share the recording, and it will also include a link to the survey if you don't mind looking for that, because we really value your feedback and we'll use it to optimize our future events like this. And we'll also keep you posted on upcoming events if you like. For example, we have an Open Access Week webinar planned for October 29th at 9 a.m. Eastern. And we will be talking in that webinar about navigating the complexities of open access with a special focus on the author experience. So once again, welcome from all of us at Highwire. I will now hand it over to our Master of Ceremonies and panel moderator, John Sack.
Thanks, Allison. I want to frame our webinar by talking about ecosystems. And I'll propose that the ecosystem of products and services gathered around preprint servers may foretell structures, suppliers, technologies, and needs in the more complex journals ecosystem. And it may even accelerate journals' ability to foster, to move faster, to publish faster, and maybe cheaper. One of the overused words in technology right now is ecosystem. I think I've probably used it 4 times already. It's a feel-good word. Something like natural or organic are in foods. Every vendor wants to claim that they're part of or in control of an ecosystem. In tech, we hear about the Apple ecosystem, pictured here, and a lot about the Google ecosystem and also the Amazon and Android ecosystems. But we have examples of less vendor-centric ecosystems as well. Blockchain, mobile phones, streaming media services, those are ecosystems also. Basically, these ecosystems are about components, whether products, services, or companies that work together, usually based on rules laid down by some central reference point like Apple or Google. These tech ecosystems can evolve fast or slow, depending on the rulemaking ability and speed at the center. I believe that the evolution rate of an ecosystem is constrained by the interaction capabilities across the components. If a lot of varied interactions are possible, then the ecosystem will evolve faster to meet the needs of its stakeholders faster. So the ability of an ecosystem to settle and adjust its rules speeds its evolution. Perhaps the best relatively recent example of an evolving ecosystem is the web itself. Now let's think about some of the authorities and rulemaking ability in our scholarly communication ecosystem. We have large publishers making rules. We have funders like Plan S making rules. We have standards bodies like Crossref and Counter making rules. And we have other participants making rules, like ISI with impact factors and Google Scholar with indexing. In the scholarly communications ecosystem, there are a number of authorities, not at all like the Apple and Google ecosystem. Of course, there are more familiar ecosystems that are not based on a central authority. Cities, coastlines, reefs, forests, etc. These ecosystems evolve slowly. Even if we don't recognize it by name, we're familiar with the concept of a business ecosystem. It's an economic community supported by the foundation of interacting organizations and individuals, the organisms, if you will, of the business world. Over time, they co-evolve their capabilities and roles, and they tend to align themselves with directions set by one or more central companies. So not a single authority, but a very few. These companies holding leadership roles could change over time, but the function of an ecosystem leader is something important to its community because it enables members to move together. It certainly helps if that leader is trusted rather than merely big or feared. I'm particularly interested here with the evolution of ecosystems with strong centers versus those with dispersed centers. Now let's look at ecosystems a little closer to home and the topic of this webinar. Research communication tools are an ecosystem, and they don't have a center at all. Donut hole diagrams like this one abound, but they often lead us to a fallacy right at their visual center. The fallacy is that the diagram leads us to imagine that there's a center occupied by a single organism who makes decisions as a central authority, in this case, the author. Maybe if we said authors, the plural, it would be a less likely problem to lead us to the single organism fallacy. There are millions of authors, and they each control their own little research communication wheel of fortune. The lack of a strong center doesn't mean this isn't an ecosystem. In fact, most all the organic natural ecosystems lack a strong center. Think again of the forest ecosystem. It might be more accurate within the limits of my paltry graphic capabilities to depict the research tools ecosystem as a nest or a web or a forest of workflows, each centered around individual authors as the authorities making their own decision about which components to connect up. But there are also some lab-based, university-based, funder-based, or company-based authorities making and breaking connections within those author-centered ecosystems. So really, this ecosystem has multiple centers. That doesn't make for a very organized-looking graphic, but in fact, the ecosystem isn't very organized from a quick decision-making or evolution standpoint. Now, forgive me for essentially retitling the same graphic and calling it journals ecosystem, but it does make a point about the structural similarity of the two ecosystems. Research tools and journals ecosystems are very similar in structure. The journals ecosystem is a group of cooperating and competing organisms that make rules and have rules made to them or on them. Imagine each circle here, not to be a workflow of an author, but to be a journal. Each has around it a set of tools and services, suppliers, authors, subscribers, editors, staff, gathered into an ecosystem by a set of rules. And of course, there are many journals. The implicit and explicit rulemaking and thus innovation operate pretty slowly. Many people have pointed out over the last few years that articles pretty much look like they did a century or more ago. But evolution can also be driven by recognition of better or best practices. The move to online journals in the late 1990s was comparatively fast. The shift to online manuscript management systems was probably even faster. Ecosystems without a central reference point, such as the journals ecosystem, tend to process, diffuse, and incorporate innovation more slowly compared to ecosystems with a central reference point, like the Google or Apple ecosystem. The ability to respond to better and best practices, though, can be spurred by mission or business goals and needs. Key goals across the journals ecosystem are variations on publish quicker, publish cheaper, publish with more consistent quality. Online journals and online manuscript systems supported those goals, so things evolved pretty fast in those cases. So what's an example of a scholarly communication ecosystem with a central reference point and thus could test and incorporate innovation more rapidly? So this brings me to the thesis that guided us in putting together this webinar and its participants. I think that preprints and journals are different ecosystems responding to some of the same needs. They're very differently structured ecosystems, and the different structure with fewer preprint servers than journals and stronger centers in those fewer preprint ecosystems may well allow the preprint ecosystem to evolve faster. In the journal system, I mentioned that the shift to online journals was fast, but in the preprint ecosystem, especially the life sciences preprint ecosystem, the shift to preprints was even faster than online journals. And the shift in medical sciences was, well, shall we say, viral fast. Because the two ecosystems share some of the same goals and stakeholders, I think it's plausible that the preprint ecosystem is a foreshadowing of innovations that could eventually catch on in the journal system. So this is one reason why journal, editorial, and publishing groups should be interested in what's going on in the preprint ecosystem. Preprints are a type of laboratory carrying out experiments such that journals might not have to. A second reason for looking at some of the innovations in the preprint ecosystem is that they might help satisfy some of the mission or business goals for journals. I don't want to suggest that I know this is going to happen, but here's what catches my eye with some of the innovations we'll be talking about today. Many of you know that Highwire recently completed a series of 25 researcher interviews and presented a webinar on this. It was a contention of most all of the researchers that, understandably, the speed of peer review had slowed down during COVID. But just as understandably, these researchers, most of them early career, needed even faster dissemination, not slower. So they told us that about half had posted preprints and would do so again in response to slower review times. Journals' mission and business goals of publishing faster, publishing cheaper, and publishing with consistent quality might be aided by some of the tools now showing up in the preprint ecosystem. Of course, the desire of researchers for quicker peer review for their papers is no surprise to anyone on this webinar. But the fact that so many of the texts that journals are seeing now have a prior life as a preprint is a new fact of life. Suppose that this could help journals improve speed of review and maybe even save money, not just time, or at least conserve journals' most precious resources, reviewer and editor time and talent. How could that happen? Well, let me take a sidetrack into another ecosystem that has a speed problem. And this is the purchase a house ecosystem, if you will, the real estate ecosystem. The example that many of us will be familiar with in our life outside of publishing is pre-qualifying for a mortgage. Imagine if all the financial paperwork surrounding pre-qualifying for a mortgage were added to the stress of bidding on a home and then losing it out that bid to somebody whose bank was faster than your own bank. That's why pre-qualification came about. If the fusty banking industry can do it, then maybe some of the tools in today's webinar might offer journals a similar pre-qualification option. And some of the frameworks in today's webinar show a way that services performed on preprints could be used to quality assess, accredit, or badge some features of a preprint to known standards. Standards a journal's editors could accept or specify in lieu of doing the assessment themselves. So in simplified terms, imagine that a journal had an editorial checklist. And you can look at the upper of the two diagrams here. And that's the qualification step. I've labeled it in a bold italic, step number 1. Imagine that a journal had this editorial checklist and that half of the items on it could be checked off based on badges assigned to preprints. Well, that would look a little more like the diagram at the bottom half of this slide, where there's the, if you will, pre-qualification step that happens before the article is submitted while it's still a preprint. That's my unproven, but I think plausible proposition that the structure of review could change with some of the tools that we're going to look at today. Now, please take a look at the names of the panel and presenters, their positions, and the areas they work in, because the slide will not be staying up during the panel. Now, before I turn it over to John Engels and our other speakers, I want to make a comment on the speakers we've invited to this webinar. Highwire is not in this webinar picking winners in the fields of products, services, and platforms that serve editors and authors and preprints. What we have gathered here is a group of very interesting services that seem to occupy different niches and thus are more complementary than competitive. I can't tell you if they'll be successful. I can tell you that they're good speakers with some very interesting capabilities, and that together they do form part of an ecosystem that's today gathered around bioRxiv and medRxiv, and in the future, they and other services could be interacting directly or indirectly with journal editors and publishers. I'll now introduce our presenters, who are also our panelists. John Inglis, who's co-founder of bioRxiv and medRxiv. John is the executive publisher at the Cold Spring Harbor Lab. Tim Vines is a chef in the Scholarly Kitchen. In addition to his blogging, Tim is founder of DataSeer, an AI tool intended to increase author compliance with data sharing policies. Anita Bendrowski, founder of SciCrunch, brings us SiteScore and also Screen It, which we'll hear about today. Josh Nicholson, founder of Site AI, a tool that offers a quantitative and qualitative insight into how scientific publications cite each other, and is also part of ScreenIt. Rich Abdul is founder of Archivist. Archivist combines biology preprints from bioRxiv and medRxiv with data from Twitter to help readers find the papers being discussed in their field. Irache Puebla is the Associate Director of ASAP Bio Crowd Review. Helen Robertson, Community Manager at Prelights. The Prelight highlights service run by the biological community and supported by the Company of Biologists. We're so glad that these innovators agreed to join us today to present and to discuss. And now over to my friend, John Inglis.
Well, thank you very much, John. A typically thoughtful introduction to put us all on the right track for this conversation. I want to thank you, John, and Highwire for arranging this event and for the opportunity to participate in it. Just let me check that you can see my screen. Is that okay?
Yes, we can.
You can. That's good. Your comments about ecosystem reminds me to point out that all ecosystems have variation, and the preprint ecosystem is no different. There are many preprint servers, last count around 60, serving different disciplines, different countries, different communities. I can speak really only about two of them, which is the servers bioRxiv and medRxiv. And I'm speaking today on behalf of this fantastic group of colleagues who do all the work that allows me to come here and talk about it and its consequences. In terms of these servers are based at Cold Spring Harbor Laboratory, but medRxiv is managed in collaboration with colleagues from Yale University, Harlan and Joe, and BMJ, Theo and Claire. And the Cold Spring Harbor-based team that I've listed here make everything happen. So these servers are community-based. They're not-for-profit projects that are rooted in Cold Spring Harbor Laboratory, an institution that for 100 years has provided all kinds of services to the research community. Both servers are funded by the lab and also by CZI. Like all preprint servers, the content is free to post and to read, but not like all preprint servers. medRxiv and bioRxiv are publisher and journal independent, although we have relationships, as I shall show, with many, many journals and many publishers. So these are, in essence, these servers are services. They're not publications, and they're not a component of a journal submission. So at the bottom of the screen, you'll just see some basic data about these servers. bioRxiv is now very nearly 8 years old, a rather substantial number of preprints there now. Already in 2021, we've posted nearly 30,000. And there are some data about the usage of this content, which is pretty substantial. And also, as you can see at the bottom now, with some time, we're seeing a large number and rapidly increasing number of citations to bioRxiv preprints. medRxiv is only 2 years old. It was 6 months old when the pandemic hit, and that has had a dramatic effect on the progress of the server. Around 65% of the content on medRxiv relates to the pandemic. And the usage for a relatively small number of preprints, 25,000 articles, the usage is pretty substantial. So we see these servers as providing services for different categories of individuals. And one of them, of course, is the information producer, the author of the manuscript. And the kind of service that we deliver for that group is, of course, speed of dissemination, as John referred to, but also a variety of other controls that the authors maintain over their manuscript. And I won't go through all of these, they're rather self-evident, but it also, I want to point out that that level of control also applies to whether or not that manuscript is going to be submitted to a journal for publication. So among the other services are a citable DOI, which is a wonderful thing for a young researcher to have when applying for a job or applying for a grant. We do a great deal to encourage community feedback by having comments on the site, engaging with on Twitter in particular. We provide altmetrics, download data, and in particular, we have arrangements with an increasingly large number of journals and publishers for the simple transfer of a manuscript to a journal for the opportunity to be considered for publication. And the numbers are growing for both bioRxiv. The numbers of journal options are growing for both bioRxiv and medRxiv, and are set to go substantially more over the next few months. And when that manuscript is published in a journal, we make a link to the published version. So I've gone too far ahead. We also provide services that we feel are directed towards readers, to information consumers. And part of that is the screening process that goes into every posted manuscript. And again, I'm not going to take time to go through all of these different elements, but the first 1, 2, 3, 4, 5 are in common across the servers, part of the screening process. The latter 4 are elements that are particular to medRxiv and we've introduced those because of concerns about the safety of medically related information that goes out to the public. We might discuss screening in more detail in the Q&A. In addition, we have the opportunity that is offered to publishers and journal editors if they wish to provide a service to their authors by encouraging them or committing them to post a preprint. Then there is the opportunity to simultaneously deposit the journal, to deposit the manuscript on the server at the same time as it is submitted to the journal. We've recently introduced a new function, which is the ability to sign up to track an individual preprint that a reader is interested in and be notified when that preprint goes through a new version or has comments or is eventually published. And the variety of discovery tools that are offered, feeds, Twitter sub-accounts, keyword alerts. One can create channels of preprints collected by different kinds of theme, particular topics, particular projects or institutions. We also have repositories for text and data mining. And another service for which we will focus on in this session is the aggregation of the evaluations that are made of that preprint in various ways by various kinds of organizations. So I've talked about the server-dependent services, but really we're here to talk about server-independent services, as John has indicated. And there are an increasing variety of these. They provide functions that the servers themselves do not, either because we don't have the expertise or the resources to do that, but mostly because we are quite clear that certain things are within the mission of a preprint server and other things are not. So we are delighted to partner with other entities that provide that kind of expertise and additional functionality. And in many ways, these services benefit both the producers and the consumers of preprints. Many of these services revolve around the assessment of the preprint, the evaluation of it, and some of it extends into the realm of formal peer review. And there's no question that that kind of evaluative process has become much more important in an era when there's been this explosion of pandemic-related preprints, and people really want to know what is important, what might be wrong, what might be clinically actionable. Some of these services are initiated by the authors, others are not. And if there is a desire on the part of the author to engage with such a service, then we have made that possible through a process we refer to as B2X, meaning from bioRxiv to another entity. These services usually provide public outputs that can be assessed by readers, and we have assembled these outputs in a dashboard alongside various other kinds of reactions to preprints, particularly media coverage and tweets. And we post on bioRxiv itself, we post the text if the authors have opted in. Otherwise, we create links to the content that is located elsewhere. So this is what that dashboard looks like. There is a series of icons underneath the author list on the left. Clicking on one of the icons that has content in it opens up the whole dashboard, and here you can see the peer reviews that appear under this project that we call TRIP, for transparent review in preprints. So there are, as I said, an increasing number of projects involving independent reviews and evaluations. In this session, we have been selective about the projects that have been invited, but all of these are worth paying attention to. eLife has adopted a policy in which it will now only peer review manuscripts that have appeared on a preprint server. Review Commons is probably very familiar to this audience. Embo Press have been posting their peer reviews of manuscripts that began on bioRxiv. Gigabyte, likewise, peer community, and a relatively new project amongst biophysicists called CoLab. These are all author-solicited preprint peer review that will be familiar to this audience. There are other kinds of independent commentary that come from projects like Pre-Review, the Johns Hopkins Novel Coronavirus Research Consortium, the MIT Press Rapid Reviews project, and the Oxford Mount Sinai Preprint Journal Club, the last two being focused very largely on pandemic-related preprints. So you are going to hear more about these, this kind of process from some very interesting, often new startups. And I look forward to hearing what they have to say in the course of the next hour or so. Thanks very much.
Thank you, John. Tim, you're next.
Brilliant. Yes, thank you. Thank you, John. Okay, I am Tim Vines from DataSeer. I also, in a previous life, used to be managing editor of a journal called Molecular Ecology. I've been in and around peer review now for about 15 years. And it's part of those experiences that led me to founding DataSeer. So I want to start with a bit of a broad context, and then we're going to sort of home in on the details. So here's a bit of a bold statement: open data equals good. You can put is or are in place of the equals as you see fit. But why? Why are open data good? Why do people care that the data sets are open? And I think it's worth promulgating this, because then that allows us to prioritize whether or not this is something that's very urgent for the community to sort out, or somewhat urgent, or perhaps not a priority at all. And here is one of the reasons. This is what I think a very compelling reason why open data is very important. This is from a Pew study back in 2019, where they asked Americans, what would make you trust a scientific article? And almost 60% said if I know that the data are available, I'm going to trust that article. And that's not because the author said the people have any intention of going to reanalyze the data or anything like that. They know that because they know that somebody can, having the data available boosts trust by a huge amount. And some of you have probably noticed that their trust in science is a major problem at the moment. So open data, if we can achieve it, is a quick win. And God knows we need some quick wins in science at the moment. And this actually also applies to researchers. This is a Center for Open Science study where they asked authors of researchers about preprints, what would make you trust a particular preprint? And here they all came down in favor of open science answers. So is there a link to the available materials, links to available study data, links to available analysis scripts? That is, if I can access details about the manuscript, the underlying materials, I'm going to trust that manuscript that much more. And it's great to have this done on preprints because then they didn't have the trappings of a journal around them, so that sort of won't muddy the picture of why you would trust it or not trust it. So this is another very important result. Open data makes researchers trust science. And that's key because scientists build their research on top of previous research. And if they don't trust previous research, then that process is like trying to jump out of a deep, muddy puddle. You can't really get very high. Whereas if they do trust it, they're trying to jump off of a solid platform, and they can get that much further and make that much more progress. So open data is good, but why? And there's another reason. This is a result I'm really excited about. This comes from Elias Berberi and Dominic Roche, who very kindly allowed me to share this. I think this is one of the first few times that this graph has appeared. So what they did was they studied a lot of manuscripts in ecology and evolution, and then they looked at the distribution of retractions. And you can see here in panel D, once journals brought in a mandatory open data statement, then mandatory open data, so all the articles were being published alongside their data, the proportion of, or the number of retractions went up really quite massively, whereas in none of the other categories had that effect. And this speaks to me to the fact that the presence of the data allows for much deeper examination of the article. And from there, we can perhaps infer that if the data are available, the research that's getting published and getting out there is passing through a much finer net. And the stuff that looks great in text, but is based on garbage data, is not going to get out there. Or it's getting out there as this figure shows, it's getting out there and then it's getting caught afterwards. If we can bring that forward and have peer review of data so that articles that are based on garbage data don't get published or don't get picked up, then we're going to be in a much better place than we are now, where you can say anything you like in the article and no one can really follow up because they can't get in at the data and work out whether you're making it up or exaggerating or cherry picking. So open data are best, to my mind, when they're part of peer review. When we move finally to a world when a journal editor can reject a manuscript, give an editorial rejection of a new submission because the underlying data sets are a mess, then we've moved from our current situation where authors can have appalling data management practices and then tidy it all up and cover it all up when they publish because the data aren't available, to a world where if you don't have quality data management, then you can't publish. And that would be a massive shift to my mind, and perhaps one of the most important things we can do. So how do we get to this point? And here's another, I'll leave it to you to work out the acronym. Here's another sort of counterpoint. Open data are PITA. Most authors don't want to share their data. They either don't have time, they don't feel like they want the scrutiny of their article. They would far rather be publishing their next manuscript to ensure that they get a grant than messing around trying to tidy up the files and work out what they did 2 years ago with the paper that's just been accepted for publication or that they're just submitting. They really don't want to do this. And if that's the big signal that the academic community has given stakeholders over the last decade of trying to promote open data, we don't want to do this. You can provide us with repositories, you can bring in policies, you can do education seminars. Very little is going to move the needle in a big way in terms of encouraging them to share their data. It has happened. It's a cultural shift in some areas like genomics, but even there has to be a fair amount of stick with the carrot. And so we have to overcome this passive resistance or complete resistance if we're going to get to the place where most authors share almost all of their data sets. So someone else needs to make it happen. And accountability is the big part of the problem here. So here is a sort of doom loop of where we are often with open data policies. Stakeholder brings in an open data policy, you must share the data associated with your manuscript. And author's like, really? Okay, fine, I'll just do a little bit. Or I won't bother with this one, but I'll do it on the next one. And so they don't really share any data, maybe one of their files. And the stakeholder then can't really check. They don't have the resources to go through every manuscript and work out what should have been done. And so they can't follow up with the authors. And so there's no consequences. And then next time the authors are told, oh, you must share your data, like, good one. And they just carry on not sharing their data. So this is a downward spiral of cynicism about open data policies because there's no enforcement. And so we need to get to the point where there's accountability. We need to specify what data sets should be shared. And so here's the opposite of a doom loop, virtuous cycle, where we have to tell authors we have a policy, but then we get to the specifics here and we tell authors which data sets from their article they need to share. And then they share them. And then there's a check that says, okay, we had a list of what you should do, and we've got a checklist of what you have done. We compare them and we can see that you've done everything. So we can go back to the authors and say, okay, you've not shared this one data set you need to share, please go ahead and do that. And at the end of that process, we then inform the stakeholder, yes, they finished, they shared all their data sets. And that means the next time the authors come to submitting an article covered by this stakeholder's policy, they know that they're going to have to do it properly. And so we enter this virtuous cycle. And really only AI can provide a scalable solution in this case. It's too difficult for people to go through every manuscript and work out which data sets the author should have shared for all 2 million odd manuscripts we publish each year. So here's where AI can come in. This is a pretty much a slam dunk for natural language processing, because you can teach a natural language processing algorithm to spot sentences where the authors have collected data. So here's a classic data collection sentence: weight and height were measured with an auto-anthropometer, and that created a data set. And here's a sentence where they describe data sharing. All the raw data, including the expressed gene lists and differentially expressed genes, were put onto the Dryad digital repository. So they've shared data. And now we just need to compare the two. And so we generate a report, and this hits, so this is what DataSeer does. We use this to generate a data sharing report. And this hits this point in the cycle here. We tell authors what they need to do. So here's a part of a data sharing report. We give them the metadata, and then we list out the data sets that we see as being associated with a particular article. And we give them the type of data we think each one is. I should point out that 80% of data sets are tabular data, and then the remaining 20% are typically the output of big machines. And we need to be specific about what the authors should do. And so here we give some specific sharing guidelines. We have a wiki where we specify for particular data types what they need to do, and we specify also the most suitable repository. We are developing a database which takes the author's context. They are funded by this funder, they're trying to publish in this particular journal, and on the basis of that information, recommend the most suitable repository for that data set. And then we need to certify that they've done it. So we tell them what you need to do. Now they check that they've done it. And so this is the sort of stakeholder informed part of the process. And this is an example DataSeer certificate for the article I was just showing, which says that they have shared all of their data sets in the right places. But we're talking about preprints today, and I want to be clear that this really isn't a cycle as such. And we can do all these steps before the stakeholder is involved. So here is a different version where we have the authors posting a preprint. And here is where DataSeer comes in to tell the authors which data sets they need to share. They go through the data sharing process, we check it, we correct any issues, and then the certification can be passed to any one of these stakeholders at a later point in the process. If they submit it to a journal, we can pass them a certificate. If they submit it, their institution wants to do a review, the authors can provide a certificate that they have shared their data, or they can tell their data, or they can tell their funder that they have indeed shared all their data sets. And so by taking open data certification upstream, everybody can benefit. That means the data are available for peer review when the article arrives at a journal. The data are available even after that for post-publication assessment. And then there's data sets available for people to do new things with, test new hypotheses. These are all the sort of very standard benefits of open data. And now, I mean, I want to, right at the end here, flick to some heavier questions. Like I established a moment ago, sharing your data is PITA, and then most authors don't want to do it. And so once they've arrived on a preprint server, will they really opt in to do something they don't really want to do? And the answer is yes, they will if their stakeholder tells them to. And I want to throw in the example of the wonderful Aligning Science Across Parkinson's, who have a extraordinarily advanced open data policy where every article by every research team in every funded research team has to go through a very detailed DataSeer open science check before the authors are allowed to submit it to a manuscript. So they post on bioRxiv or even in upstream in a private folder, the article's assessed, we check all the aspects of open science, and then the authors carry on from there to submission to a journal, having shared all of their data, all of their code, made sure all of their materials are correctly cited, and so on. But who pays for this? Getting to this open data is not easy. This is why authors don't want to do it, why stakeholders don't currently do it. How are we going to make this happen? How are we going to make this work long term? How are we going to make it worthwhile for people to do this, for us to do this? Should it be free for the authors? Charging authors to do anything is often, unless they're getting a big payback in return, is not really a viable strategy. So which stakeholders value this most, and who's going to get involved? So those are my questions. And also one last point is that if we are doing certification upstream, then it's not clear which stakeholder, because any of them could benefit from this, which of them should enforce or promote obtaining these open data certificates. So I want to stop there. Thank you very much.
Thanks, Tim. Anita, on to you.
Thank you very much. And all right, I think hopefully everybody can see that. All righty, so my name is Anita Bandrowski, and I am from SiteScore. So this is a tool that, oh, here we go, can review any kind of scientific paper content. And so one of the questions is, how fast can we review? What can we review? And then, of course, there are questions about quality of review, right? So this is the set of algorithms currently. It's at 55 different algorithms that check against various rigor criteria guidelines. And here is just a list of these guidelines. This is a little bit too much for us to go deeply into this, but we're covering about 85% of the MDAR required issues at this point, and the tool also creates an MDAR report automatically. All of this happens very quickly. It happens within seconds. The journal submission platform basically asks the authors to copy and paste their methods into a question that goes to an API, and nearly immediately there is a set of reports, including the star table for cell press journals, the core report, which is what I'll show you in a second, and the same information essentially is also reformatted into an MDAR report. All of that happens essentially immediately. So this is what a submission system integrated platform actually looks like. We're integrated with both eJournal Press and Editorial Manager. Here is that extra question. There could be a submission button here, submit directly and iterate, dear author, or you can submit with the, with the author pushing either submit to journal or submit revision. An email is generated. They do get a numerical score. I'll show you a little bit more about that. And they also get this very lovely report. So here's what the report looks like. This is, again, automated. And you'll see in this report, there are blue things and there are red things. And let me just highlight some of those. So here is one of the bits of that's kind of easy to understand. I think this is a GitHub repository that's being cited in this sentence. Here's the sentence from the author. And what the code does is it goes out to GitHub and it says, hey, are you a live page or are you a dead page? Here's an example of when it comes back live. Here's an example of when it comes back dead. When this page is dead, right, basically, this could be the author has not made that repository public, or they've made a typo in the URL or some such. Here are typos in the identifiers for some data. So, SiteScore also checks for data information, but we're certainly not the only AI tool that does that. And actually, what I found with some of the other tools when looking at the data information that each of the tools actually reaches into the papers for, they are often quite complementary. They're not actually, nominally they're asking for the same thing, but in fact, the more the tools, it seems, the better. Here's another bit. This checks for clinical trial identifiers, and this little warning basically says, so the blue says, yes, this is a valid clinicaltrials.gov number or EU Clinical Trials Register. This is a warning that this clinical trial is actually registered in 2020, but the trial was started in 2012. So, that doesn't do anything to the report, but it's a nice little thing that often journals have a hard time checking by hand. In the cases where a rigor criteria is met by some sentence in the paper, that sentence is brought into the report. Otherwise, it is either not detected or not required. Those are the two options for when the sentence is not present. And that just means that the tool thinks that this particular piece of the, of the larger checklist is not really required for that particular type of paper. For reagents, we have kind of three states. So here's an antibody sentence that this author has used. It pulled out, the tool has pulled out some antibodies, but there is no identification information about this particular reagent. In this case, there is a suggested RRID for this one. Because this is gene expression omnibus, that was sufficient information for our tool to then suggest an RRID for this, this particular database in this case. And here's a third case where the authors actually did use an RRID, and that's simply verified. So in cases where cell line RRIDs are used, we also have a warning for all of the misidentified and misclassified cell lines. So that warning comes up right here. And currently, we're not doing a whole lot with the statistical tests, but we are pulling them out, and we're working on doing more things with those. Each of the reports also receives a score between 1 and 10. And of course, 10 is better, 1 is worse. but it is kind of difficult to know, and I'll show you a little bit about this. So RRIDs, again, it's a no-brainer when you ask people to identify reagents inside of their papers. Then the reagents are more are able to be found more easily. That's all that this says. Basically, we go from our ability to identify reagents at about 50% rate to about 90% rate in the papers that use RIDs. Not surprising. But what's a little bit more surprising is that when you have these bad reagents, like these contaminated or problematic cell lines, there's a warning when the author has to go through and find their RID, that warning that I showed you with SiteScore. And that actually produces a 66% drop in the usage of these reagents. And that's with no additional editorial instructions or oversight. So this has been a very interesting positive benefit out of all of this. So now the question is, what are these numbers? What do they really mean? And this is a part of a study that we published in 2020, showing the average numbers across this particular 10-year period. So this is this black line right here. And you can see that the average number went from a little below 4 to about 4.2 or so, give or take. And this is the average score across about 2 million manuscripts in PubMed Central. And superimposed upon that is the same score for the Nature Journal. And so here is this light blue line, and this is an average of all of these different criteria. So basically, you could see that their average score before the implementation of the Nature checklist was much lower than the average of all journals. And then after the checklist, it actually got a little bit higher than the average score across all journals. And the reason that happened is because largely now Nature authors were addressing things like the randomization of subjects into groups, sex as a biological variable, antibody findability, blinding, and other things. Organism, actually, they never really pushed, and it kind of seems a little flat over this 10 years. Hopefully that will improve soon. Okay, and because there's a bunch of publishers in the room, I had to put in this slide. I'm sorry. There is absolutely no relationship between this metric of quality of the methods section and the journal impact factor. So we'll just stop that at this point. But this quality metric does improve if you continue to use the tool. This is data presented a couple of months ago by AACR, showing that at initial submission, the SiteScore value across all of their journals was quite a bit lower versus at first revision, it ended up being a little bit higher. And at second revision and then publication, it will be even higher. So on average, the authors are doing things to address some of these rigor items within the review process. We're still not sure whether the running of SiteScore has a whole lot to do with this, but at this point, this is the thing that we have. But this graphic actually shows, another one of these outcome measures, the inclusion of RIDs in published articles at AACR. And you can see we're here in March of 18, AACR asked very nicely that authors include RIDs, and largely they didn't care. But once AACR actually implemented a letter to authors, now many more of the authors started to care. And more recently, in March of 2021, they've actually started to ask for a higher SiteScore in order to actually publish higher than 4. So we'll hopefully see what happens after that point. We're still waiting on those results. Okay, and of course, I am a tool maker and SiteScore is my tool, but my tool, and there are many other tools, and you'll hear from Josh in a second with Site, but a lot of us actually got together during the pandemic, and we said, hey, why don't we actually take all of these tools, put them together in a single pipeline and run them across all of the COVID-19 literature, because it's a pandemic and because we're going to take whatever blowback that we get, and there was a little bit, especially in the very beginning, for the good of sort of humanity, right? So we brought together things like OddPub, which does check for the presence of open data and code, Limitation Recognizer, Bazooka, this screens for bar graphs of continuous data, JetFighter screens for rainbow color maps. So that's for colorblind individuals, figures that are inaccessible for them. Trial identifier searches for clinical trial numbers, are transparent, reports things like conflict of interest, funding, and registration statements. And we started out with Seek and Blasted, which checks for incorrectly identified nucleotide sequences. However, this tool is semi-automated, so they have not quite been able to keep up with the volume of COVID-19 preprints. We did publish a lovely manuscript earlier in the year talking about some of the results and the quality of data that's coming through the preprint servers. This is what the pipeline looked like. So basically, we pick up, thanks to the nice people at bioRxiv and medRxiv and the very liberal licensing of this content, we are able to pick up this information. We're able to identify the COVID-19 preprints. We are then able to run that through a series of parsers, some for text and some for images. And then we run those sections of the manuscript through the various tools that recognize and report upon either the images, the image quality, or certain parts of the text quality, and of course, reference check for Josh's tool. Everything gets put together into a single report. That report is posted to a special group. Thanks to bioRxiv, we have our own special group in Hypothesis. And of course, we tweet about this because at the time that we started this, Bio2X or the R2X or whatever that piece wasn't quite available yet. So the only way to let people know that these reports exist was by using Twitter. So that's kind of what this looks like. And what you can see here is that we've scored about 19,000 preprints. We do this every day. So if you go to the SiteScore reports webpage, you'll actually see, the current number of tweets. There's basically one tweet per paper. And so that's all accessible from the little Twitter tab. This is what a tweet looks like. And the tweet links both to the preprint and it links to the report. And what you can see in this report, I hope, is that SiteScore provides part of that report. And then there is a set of results from each of the tools going all the way down, talking about, what is the, if there is a particular problem that the author needs to be alerted to. So this really shows that we can, in fact, review preprints extraordinarily quickly. I don't have a lot of time in terms of how this was received, but I will tell you that currently we haven't had, knock on wood, a whole lot of people complaining. And I think that's probably because people have started to get used to seeing these little automated reports coming up. That was not the case when we first started. There were a lot more people complaining, some of them to the founders of bioRxiv, but it is what it is. But we can review preprints, and we do so without a lot of input from human beings, which means that we can keep up with the literature almost at any volume. We can review lots of different parts of the literature, and the quality is always consistent. So that's a very nice thing. It is not 100% accurate, but it is at least extraordinarily consistent across all manuscripts. And it does not matter that the scale at which we are reporting, because a lot of these tools have already been run across millions of manuscripts. We even have very good standard metrics for what the tools will actually provide. Okay, so I just want to say thank you on behalf of myself and behalf of the Automated Screening Working Group, which is now has been rebranded to Screen It. And I encourage you all to take a look at the Twitter tab of your favorite COVID preprints. Thank you.
Thanks, Anita. Just before I turn this over to Josh next, I wanted to mention that there is a Q&A button, which is capturing a number of questions from the attendees, and they're being answered by the panelists as we go forward. So if you have open questions or comments for the panelists, please go ahead and put something in the Q&A panel. And over to Josh.
Great. Hello, everyone. Let me share my screen and get this up. So my name is Josh Nicholson, and I'm the co-founder and CEO of Site. I'm excited to be talking about preprint, something near and dear to my heart, as I launched my own kind of preprint server, about 6 years ago, called the Winnower, which was around the time bioRxiv and medRxiv was launching, and we focused a lot on publishing gray literature, so the things that are outside of the scope of a traditional article. And so in some senses, my learnings from that has contributed to what we've built at Site. And so I have a background in biology, cell biology. I finished a PhD about 6 years ago, and that also contributes to how we're building this tool. And so I like to start with my talks by just kind of reminding us that all these things we're dealing with, publishing, AI tools, impact factors, editing, et cetera, behind that is really amazing, work. And I've been showing this slide for over a year now, and I'm going to continue to show it because it starts to, emerge and become very self-evident. And so pre-pandemic, I was showing this, and now with life-saving vaccines, I'm excited to continue to show it. And so this allows us, this research allows us to have virtual conferences like this. It allows us to get back to kind of quote unquote normal life, the powerful computer in your pocket or purse. And all these things were ultimately informed by basic research, applied research. And I think it's good to step back and kind of acknowledge that. At the same time, obviously, and we wouldn't be building tools if we didn't acknowledge this, there's challenges, right? So there's a massive amount of content, especially in preprints. John talked about this explosion in preprints. And now with that kind of impacting our day-to-day lives, how do we know what to read? How do we know what to trust? And that's really kind of at the mission of Site, is that we built Site to make research not just necessarily more impactful, but more reliable, to make it more understandable, and to make it more nuanced and provide some context. Because there's a conversation happening amongst papers, amongst preprints, that is lost in our typical superficial way that we count citations. And so Site is really kind of trying to bring in the next generation of citations to change the conversation from how many times has this been cited, whether that's a preprint or a server, to how has it been cited. And we do that by showing the in-text citation, whether that's from an article or a preprint, classifying it. Does it provide supporting or contrasting evidence? Does it come from the results section or discussion section? And then allowing people to kind of see this conversation very easily. So moving beyond just counting citations. And so to date, we have analyzed over 26 million full text articles, as well as preprints, and we have extracted and analyzed 910 million citation statements. And so what does this allow you to do? And so generally, when we look at a research publication, whether that's a published article or a preprint, we generally look at a few things. Where is it published? Who are the authors? What are their affiliations? And then some metrics. And I think in all these cases, we generally ask, do I know these people? Do I trust this? What is the reputation of this? And with preprints, there's some reputational cues that you lose out on, right? You may not know these authors, and I think that can be a good thing as well as a bad thing. It really kind of sets the stage and level sets research publications to force you to think critically, is this something that I can trust? And obviously, in a perfect world, we read everything and really go deep into it. But this is not, a perfect world. And so oftentimes we do look for cues. And I think a lot of these tools that we're starting to build will help, assess and kind of guide readers to, this is something you can trust. This is something maybe, untrustworthy. And so we have built this browser extension, which is free and works for Chrome, Firefox, Safari, and even Microsoft Edge. And it will show you our information virtually anywhere you're reading it online, including bioRxiv and medRxiv. We also now have a badge live on Archive, not requiring you to have the browser extension. And what it shows you, if you click, is the conversation happening amongst papers or preprints. And so in this case, you can see that it had 52 citations. Most people are not going to open 52 papers just because that's a massive amount of work. But now using Cite, they can very easily browse what have the citing papers said. And in this case, they show that there's some contrasting findings. So opening up this conversation, gets back to one of the aims of Cite is to provide this more context and this nuance, because it's not just always highly cited is good. Maybe some, are calling this into question, or maybe others have found similar results, or maybe they provide important interpretation and nuance and understanding. And so the mentioning bucket of citations is also extremely valuable. And so how does this, more directly impact preprints? And so there's, constantly this debate about preprints are bad, preprints are good. And I think, this is oversimplified, but there can be challenges. And so here I'm showing, an example of a preprint that received a ton of media attention. So it had one of the highest altmetric scores, top 5% of all research by altmetric. It was shared across many news outlets, many blogs, and very widely across Twitter. Right. So what would they say about it? You can maybe read through some of these tweets, and oftentimes there's very valuable information on Twitter, but sometimes this is too much. And so in this case, here's this medRxiv preprint. It was highly popularized. It was subsequently challenged, and interestingly, it was subsequently challenged by another preprint. And so preprints have this self-correcting mechanism, this way to cite that is often much, much quicker than, a paper citing a preprint. And so we have, looked at citation rates and citation counts in preprints, and there can be within days, because of the rapid process of posting a preprint, a supporting or contrasting citation. And so it's speeding up this conversation and making it, instead of sending kind of snail mail, you're now sending emails, if you will continue with that analogy. And so what about a new preprint that hasn't received any citations? And so here is looking at a tweet from September 29th by Eric Topol, the head of Medscape, showing this image from a preprint that makes a pretty big claim, saying that there's no significant difference in viral load between vaccinated and unvaccinated, asymptomatic and symptomatic groups infected with COVID. And so I, looked at this not from a professional standpoint, but from a personal standpoint. This is something that may lead people to get, less vaccinations. This should be really something that we think is strong if we're to broadcast this, or really, what does the literature say about this? And so obviously this preprint hasn't gotten any citations at that point because it was just posted. And so one thing that our citation statements allow you to do is to search, key claims, key topics, key reagents, really any word, to see what does the scientific literature say about that. And so using our browser extension, you can right click and highlight anything anywhere you're reading online to see what does the scientific literature say, including the preprint literature say about this. And so if you do that, you come to our citation statement search, and what it shows you is this paper or this preprint, also from medRxiv in 2021, documenting a different finding. And so they're saying that there is a difference between vaccinated healthcare workers and unvaccinated healthcare workers when you're actually looking at, culturing this. And so not just, RT-PCR. Going down, you can see that this has been supported. And so if you click that, you can again see this conversation opening up amongst this massive ecosystem of preprints, where another group has found similar findings where there's a difference between these groups. And so this information's there. It's just hard to navigate your way through it because there's so much information out there. And so that is what Cite is trying to help with, is to show this context, to show this conversation happening amongst preprints. And I think that is useful whether you're a reader, an editor, an author as you read, because you can start to see things that are out there that are surfaced for you to explore. And so one thing that Anita talked about, which I'll look at a little bit and pick upon, a publication, is this reference check. And so here is an example of a paper that was published in November 2020 in Frontiers in Psychology. It doesn't have any citations. And so we have developed a tool or part of our tool called the reference check, which helps authors, and this is now working with preprint servers, to look at the references that they are citing to make sure that they're not retracted, to make, or more generally, to see how they've been cited. And so if you take that PDF, whether that's an unpublished document, a preprint, or a published document, you can upload that to our reference check, and we're also integrating this into submission systems. And what this will show you is how your references have been cited by others. And so at the top is a paper that was published in 2004, was actually retracted in 2013. And so that paper is citing a reference that has been retracted for 7 years. And this is, not a live shot, but you can expand that and see that they're unaware of that. And so this continually happens across publishers, across journals, even in preprints, because it's hard to say, what I read now or years ago, how has that changed? And so we're trying to help authors look at this at the point when it's not too late, right? You can't remove this citation. I mean, maybe you can correct this, but at a preprint stage, you can easily change that. And so in this case, this is pretty, clear cut. This should not happen. If you scroll down, you can see who else has cited work that I've seen. And so maybe looking at this reference, you can see there's 3 supporting citations and 2 contrasting. This doesn't mean you shouldn't use this reference, but it's a quick check to see what these contrasting citations have said, or these supporting citations have said, or more generally what others have said. And maybe that will help with your interpretation, how you cite things, and maybe, give a more full picture. So this is now integrated into major submission systems, and I think would be a key, integration into preprint servers to help authors, make sure that their foundation of their paper, if you will, is sound. And so this is the last slide, but I like to kind of acknowledge, or to cite, if you will, that this idea of adding, markers to citation indices has been around a while. It's just been technically very hard to do. And so I think we're lucky to be, text mining papers because there's enough open ones of it, and because the tools are there to do it, to actually now start to build this and, hopefully bring in kind of the next generation of citations. And so I would encourage, any and all questions as well as feedback, hopefully during the panel or on Zoom, and I can look at that. And then you can also email us or email me directly, as well as tweet at us. So thanks so much.
Thanks, Josh. Thank you.
Sounds good. Okay, and you can see my slides okay. Great, okay, everyone's muted. My name is Rich Abdil. Oh, great, okay. My name is Rich Abdil. I'm a PhD candidate at the University of Minnesota, and I'm the developer of archivist.org. Before we get started, I should also disclose that I work for Dimensions, a company that provides bibliometrics data and services to organizations, including publishers whom I assure you I have not been allowed to talk to. None of the information today uses Dimensions data directly, and they have no idea what I'm going to say. So the archivist.org project is the most visible component of the work I've been doing. It basically works as an alternative front page for bioarchive.org. We index all the publications from bioRxiv and medRxiv, including monthly download metrics, and combine that with Twitter activity. The idea was to provide a way of sorting new preprints by something other than chronology. So we tried to find an obtainable measure of discussion activity somehow. You're welcome to use our website, the API, or the database snapshots for your work. All the information is on the website there. But I think for the group today, which I think is mostly people related to publishing, I think it's more of an example of what you can implement at your organization if you want to. Archivist is not a particularly sophisticated application. It combines data from 2 sources into 1 source and generates a web page. It's playing the piano with a sledgehammer. bioRxiv has an API now, and other preprint servers have programmatic ways of accessing their information. So if your organization has web developers or data scientists, it's never been easier to build your own integration or combine existing services like the ones we've been talking about today that you really like. And as for the archivist data, the interesting side effect of displaying this kind of information is that we also have data on every bioRxiv preprint. And the reason that may be relevant to this group here today is because, at least partly, because of the ways preprints could be integrated into publishing workflows, particularly as a source of potential submissions. This itself isn't a new observation on my part, but the data we've collected provides some useful context that I'd like to share today. The first general observation is that there are a lot of preprints. I won't spend too much time on it here, but almost every field is growing exponentially. This graph cuts off at the end of 2018, and things have leveled off a little bit since then for bioRxiv specifically, but there are a lot of fields that are still growing very, very quickly. The top graph here shows growth in number of downloads in each month. That's growing pretty consistently. And there are tens of thousands of preprints that aren't included in this graph from websites like arXiv and Research Square and OSF preprints and so on. We can also see who's publishing them. In general, more prolific journals like Scientific Reports and PLOS One tend to publish more preprints, but proportionally, there are still some standouts. Journals like GigaScience, Genome Biology, eLife, Nature Methods. In recent years, we found that each of those journals had at least 30% of their published papers also available on bioRxiv. And for GigaScience, it was actually almost 50%. So there's a lot of variation, and some of that might be happening afterward or as part of the submission process, but it seems like there have been journals looking through preprint servers for a while already. And once we know where preprints end up, the fun thing to do next is to pit them against each other. So here's a box and whisker plot without the whiskers, just for readability. And it shows the average downloads per preprint that later appeared in each of these journals. So at the top, we can see Nature Methods received, the median manuscript published in Nature Methods received about 2,400 downloads as a preprint. And when we compare this to the median of about 260 downloads for preprints published in PLOS One, we can start to see the trend depicted in the inset here, which shows that journals with higher impact factors tend to publish preprints with more downloads. I could probably fill the rest of my time with the caveats to this assertion, what impact factor measures, what downloads might measure, what median downloads might measure, which factors are confounded with which ones. But independent of the debate about how to operationalize popularity, I think the main lesson from this figure is that different journals are already taking different approaches to preprints, and those differences have consequences. Editors have a growing number of tools to evaluate preprints that are flowing into life sciences preprint repositories. Some of them we've heard about today, some we'll still hear about, and I'm sure there's going to be more coming down the pipe in the future. But if one of the goals of integrating preprints into your publication workflow is to try to find interesting work by people who aren't already submitting to you, the process may have to be more intentional than that. Last summer, we evaluated authorship in bioRxiv preprints to quantify where they were coming from, basically. And the answer for the most part is the United States, followed by the UK, Germany, and France. However, different countries have very different levels of research. So we also evaluated bioRxiv adoption as a function of total scholarly output. In this scatter plot, each point is a country. The x-axis, along the horizontal axis here, is total citable documents published between 2014 and 2019. And the y-axis is total preprints on bioRxiv. So countries along this big red line, or countries above the big red line, are overrepresented in bioRxiv relative to their overall output. So countries like Israel, Switzerland, the UK, and way up in the top right corner is the United States. Countries below the red line, for any number of reasons, are underrepresented on bioRxiv. The most dramatic differences are illustrated in this bar chart on the right. So Turkey has the lowest representation relative to their overall academic output. Other countries like Iran, Malaysia, Greece are also on the list. China, despite having lots of preprints over here, is also underrepresented just because of their very high overall output. They have almost as many publications as the United States, but far fewer preprints. This is a really busy slide, but the point is that because of the outsized presence of countries like the US and UK, most countries in the world are underrepresented on bioRxiv relative to their overall scientific output. And we found another interesting dynamic when looking at the proportions of published preprints that come from each country. The idea was that if 10% of published preprints come from country X, we'd expect 10% of preprints published by any particular journal to also come from country X. And that definitely was not the case. So even if we ignore overall research output, differing publication rates between countries, differing patterns of collaboration, everything that gives the United States a leg up in this equation, even then, there are journals that publish more US preprints than you'd expect. In some cases, almost twice as many as you'd expect up in these journals at the top here. And my point isn't to criticize bioRxiv or any particular journal or publication or system. It's only to point out that these systems are changing quickly, and that's really exciting. But when we encounter new models of sharing research or evaluating it or reviewing it, the early adopters are going to look a lot different than the rest of the world, the people who aren't in a position to experiment for whatever reason. So the growing preprint ecosystem we're talking about today is an incredible opportunity to democratize scientific research, to try leveling a very lumpy playing field. But jumping into it from the publishing side requires going into it with some intention, I think. And I'm bumping up against my time here. So I just wanted to thank you. The publications I've been discussing are on the left, and on the right side of the slide are the ways you can find out more about our research or get in touch. I appreciate your time.
Thanks very much, Rich. And now, Irache.
Yeah, thank you so much for the invitation to be here today in such good company. I'm just going to show my slides, and hopefully everything should be coming as it should. Right, I'm Irache Puebla from ASAP Bio. I'm going to be speaking about some initiatives, mostly one pilot program that we have ongoing, specifically around preprint review. For any of those who may not be familiar with ASAP Bio, we are a non-for-profit. We seek to drive transparency and innovation in science communication and have a big focus in promoting preprints, and related to that as well in the promotion of preprint feedback, so preprint commentary and reviews. And to set the scene a little bit before I speak about our project, I just wanted to mention that the fact that we've heard quite a bit about tools, etc., to provide feedback and engage with preprints. But one element that I wanted to highlight today is the fact that there has been some challenges around participation in preprint commentary, at least in a public format. We are seeing, and again, this is based on data from bioRxiv and medRxiv. Some other preprint servers may see different patterns, but a low number of preprints are getting public comments. So this doesn't mean that there is no feedback and engagement happening. There is quite a bit that takes place on social media or through email privately. But in terms of public commentary, there hasn't been a lot of participation. And when we think about the factors that may be behind this, there are a number of those. One is potentially the lack of incentives. What's in it for me if I'm a reviewer and I'm commenting on a preprint? There can also be questions about the visibility of the comments. There are different platforms. Sometimes the comments on a preprint may live on a place separate from the preprint itself. Although, as John was explaining, bioRxiv has a very nice dashboard, and I'm a great supporter of the tools they are including. There can also be concerns around whether the authors who posted the preprints actually want public feedback on their paper. They may be happy to have comments, but perhaps not in that forum. And also questions about potential vulnerability for those who comment and perhaps post critiques on preprints, especially if they are to have their names associated with the critique. So what we have done at ASAP Bio was that we wanted to explore new ways of experimenting with preprint review to see if we could encourage higher participation, particularly by early career researchers, and also making sure that there was this element of the public posting, making sure there was a public record of that engagement and feedback. And the idea for this trial really was inspired by the select crowd review model that has been pioneered by the Sinlet Journal. Benjamin List had this idea and implemented it at the journal several years ago. And the idea here is to develop a group of reviewers who are invited to comment at the same time in a short period of time on the paper, and they could provide comments on specific parts of the whole paper. And that group of reviewers is what we are calling the crowd. So we wanted to see if we could transpose that model to the space of preprint review. And with that, because we wanted to start small and see how this worked, we started with a specific discipline, which is cell biology. Again, we are looking for preprints posted on bioRxiv. At the moment, we are running at a scale, again, low scale, 1 preprint per week. And we are running this as a 3-month trial running until the end of November. So briefly, the way this works is that to start with, ASAP will contact the author of the preprint and ask if they're happy to participate. And this ties to the item I mentioned about the potential concerns of whether the author is happy to have eventually a public review on their work. So provided that the author consents to participate, we will then circulate the preprint to the crowd. So this group of reviewers who have signed up to provide comments, and we invite them to discuss the paper privately in a private hypothesis group for a period of 7 days. So essentially, that's a group that is only available to those who are participating as reviewers. After that period of time, we will synthesize the comments into a single unique review that we post publicly through bioRxiv via the TRIPS, so Transparent Review in Preprints mechanism. And like that, it is publicly available along with the preprint itself. We've been running this for 6-7 weeks now. What's our experience so far? We have had a fantastic response by researchers interested in participating in the review activities. Over 100 researchers have signed so far. We have completed the commenting period for 6 papers. In its case, we had something between 5 and 90 people commenting. They could contribute a full review or specific parts as they preferred. We posted all of those reviews, and also interestingly, there was quite a bit of interest from the authors to provide a response. We've posted 2 public responses and had some interest from other authors as well. All of the evaluations are posted publicly. As I mentioned, they are available on bioRxiv, and we are very grateful to bioRxiv because they have also prepared a collection for the evaluations from this group. And at the same time, they are also, all of these evaluations are also appearing in Society, which is a platform developed by Life, where they aggregate reviews and evaluations on preprints developed by different groups. So we are also included there. And then what I wanted to say here is perhaps thinking a little bit ahead, because I mentioned that this is a relatively short trial, but we may, there are a couple of things that we want to learn from this and again, potentially explore for the future. And I'd be interested in the attendees' thoughts and questions as well. One is that this can be a potential opportunity to expand the reviewer pools at journals. We often hear that it's very tricky to find reviewers for journal submissions. Our experience in this trial is that there was quite a bit of interest from early career researchers in participating. And we are encouraging to also take part in another initiative we have ongoing in parallel, which is called the Preprint Review and Recruitment Network. What we do here is invite researchers to share information with us about the public preprint reviews or comments they have posted. And then we will share that information with a group of participating journals. We have currently 42, so that they can use that preprint review information as work samples to consider those researchers for roles at the journal, either as part of their reviewer pools or perhaps even in roles as editors. And then again, thinking ahead, this is even farther ahead, but one of the benefits that we see in providing public preprint reviews is that it opens the door to the portability of those reviews. And I know there have been a number of experiments before, so probably we'll need to see further experimentation in this space. But we do see potential benefits for those preprint reviews in the space of the journal and the editorial process, because they can, again, help editorial decisions at the triage stage, selection of papers, complement the peer review reports commissioned by the journal, again, if there are any bottlenecks as part of that process, and sometimes even provide specialized comments for some papers, perhaps interdisciplinary or the required specialized expertise that's not always very well covered in journal review. Obviously, this is going to need some further experimentation and developments, and I'm just offering here some thoughts, and again, I would welcome comments from others. I think it would be interesting if we want to materialize this portability to ensure that there is this partnership with journals to make sure that they are happy to reuse those reviews, and that we have mechanisms to both surface the preprint reviews when they happen in real time. I think that's one of the challenges now if the editors want to use preprint reviews, is that they may already have the peer review process at the journal ongoing. So how do I need to keep up with what's happening in the preprint record every time? If there will be tools that could surface that in an easy way, as well as potentially have some mechanism to manage, again, can we gather some of the reviewer identity and expertise information, or at least do that in a way that doesn't necessarily break the anonymization, but it essentially provides some verification process so that the editors can trust the expertise of those preprint reviews. And just to finalize very briefly, in the context of portability of reviews, I also wanted to mention another initiative, which is a partnership between ACEBio and EMBO. EMBO operates the platform Review Commons, which is a platform that allows independent peer review for manuscripts before journal submission. The way this works is that the author can submit their manuscript, which they may have already posted on a preprint server, submit it to Review Commons, and this platform coordinates peer review pretty much in a similar way as the journal would do. And those reviews are then shared with the author, who is invited, if they didn't yet have a preprint, to post a preprint with the reviews, and if they feel more comfortable doing so with their own response, in the form of a refereed preprint. The advantage of Review Commons, and this is where the portability element comes into place, is that it's run in collaboration with 17 affiliate journals who have agreed to reuse the reviews provided by Review Commons, which means that the author can get their paper and the package of reviews, and the journal will be able to make an editorial decision based on that. And the idea here is really to avoid those serial re-reviewing cycles and provide, again, more transparency, but also better and faster experience for the author who is looking to have the preprint, but also the journal publication. And with that, again, we have plenty of resources on the ACEBio and Review Commons websites, and I encourage comments and questions. Thank you very much.
Thank you. Helen, and Helen is our last speaker before we head into panel.
So yeah, thank you. My name is Helen, and I am the Prelights Community Manager. Thank you to Highwire for inviting me to speak about Prelights today. I'm going to use this time to talk a little bit about the objectives of Prelights, how we go about achieving those, and then talking about how Prelights feeds into that wider community review of preprint narrative that you just heard about a little bit before, and how that fits a bit with what ASAP.bio are doing. So Prelights is a preprint highlighting community, and we're supported by the Company of Biologists, who are a not-for-profit science publishing company based in Cambridge in the UK. And you can see on the right, these are the 5 journals that the Company of Biologists publish. And I think it's worth highlighting here from this timeline, you can see how the Company of Biologists, the publishing side of that, has been involved with preprints today. And actually, since the end of 2016, all of the Company of Biologists journals were integrated with bioRxiv with the one-click submission option. So as of the end of 2016, you could simultaneously submit your manuscript to bioRxiv and to one of our journals, just with that one-click approach, which was really nice to see. And then about 3.5 years ago, so at the beginning of 2018, is when Prelights was actually launched. And Prelights was launched with 2 main objectives. So we know in the past kind of 5 or 6 years, the number of preprints that are being posted related to the biological sciences has seen a really big increase year on year, if not month on month. And that's great. It's really great to see in terms of disseminating research, but it can be very easy to get lost in that influx of manuscripts. So one of the main aims of Prelights was to provide a platform to help scientists navigate that influx of research and to highlight preprints that might be interesting to them and provide a digest of that work. The second objective of Prelights was to provide a public platform for the discussion of preprinted research. So I would say historically, and perhaps even to an extent now, a lot of the discussion that we see around preprints happens on sites such as Twitter, which is great, but it's also relatively transient. So the second objective of Prelights was to provide a platform where highlights of work could be submitted, but after they've been submitted, the authors of the preprint itself were welcome to comment on it. And of course, any readers of the Prelights website could also comment on that work and feed into that wider community discussion. And I've just included this little screenshot at the bottom. So as of summer this year, Prelights was shown under the bioRxiv community review tab, which was really great. I'm happy to see that. So with that in mind, how does Prelights work? Well, at the center of Prelights is our community of Prelighters, and our Prelighters are mostly early career researchers. So they tend to be PhD students or early career postdocs who are interested in preprinting and keeping on top of new research. And they select preprints from any preprint server. It's mostly bioRxiv, but we do sometimes highlight work posted on other servers. And they write a news and views type digest of the preprint, which is sent to the author of the preprint. And they are welcome to answer questions that the Prelighters have raised or to provide any other comments on the Prelight post before that Prelight post gets put on our website. And then once it is on our website, it's welcome that for any readers of the site to comment on the work and try and continue that discussion, as I said. So we're not prescriptive about how our Prelighters write their posts, but they do tend to sort of fit a general template. So we always have the preprint info and a link to the preprint itself at the top of the post. And we include a tweetable summary, so a kind of snappy summary of the work that we can put on Twitter to encourage visits to our site. And that's really important for Prelights. We find about 40% of visits to our site come from click-throughs from those Twitter posts. All of the Prelights posts have some background or a short summary of the context of the work, and then the main findings of the work. But the most important part, we think, is the Prelighters' opinion. So why they chose that post, why they think it's important, why the preprint might be important to the research community, and then questions to the author. And the questions to the author is, again, a really important part of the Prelights post. And we find that in the vast majority of cases, authors of preprints are really glad to see that their work has been selected to be featured on Prelights. About 2/3 of our Prelights have comments from the authors or answer the questions that the Prelighters have raised. And I've just shown on the right here, this is some screenshots from Twitter with authors showing how glad they are to see that their work has been highlighted by the Prelighters. And that's always tends to be a really positive experience for the interaction between both the Prelighters and the author of the preprints. But even further than that, you can see that in some cases, the Prelight post goes on to influence the manuscript or change the way that authors think about their work. And I think that's a really great thing to see. It's not only testament to the type of questions that our Prelighters ask, but it also shows the benefits of facilitating that discussion around preprints as a way of aiming for manuscript improvement. And it shows that we don't need to rely on that formal peer review for comments on manuscripts and thinking about how to improve them. And as you just heard with the ASAP Bio initiative, I think this all feeds into a positive view of community review or the community discussion around preprints prior to their formal publication in a journal. And as I said, at the heart of our Prelights community are our community members. So we have around 300 active Prelighters from all across the globe. And as I said, they tend to be early career researchers. We've featured Prelights covering all of the bioRxiv categories. And so far we have about 1,200 Prelights posted on our website. For the Prelighters, being part of our community is a great way to stay up to date with new preprinted research, to build their peer network outside of their lab or the immediate institute that they work in, and also to gain experience in science writing and editing. I'll also note that Prelights is indexed on various websites, including EuropeMC and SCIETY, and we also encourage all of our Prelighters to add their ORCID ID to their profile, so you can click through from our Prelighters to their ORCID profile itself to see the work that they've published or reviews they've been involved with. So this is a really great way of raising visibility, not only of Prelights, but of the Prelighters themselves. I also think that Prelights is a great entry-level approach to the peer review process, and we do view writing for Prelights as being quite different from peer review. We view it more as a community review or community discussion, but we know that there's a huge variability in when researchers become involved with the peer review process. So some researchers might be involved in grad school and others not until they might even have their own labs. And there's a huge variability as well in how much mentoring these researchers have in getting involved in the peer review process. So I think Prelights is a great way to kind of kick that off and get involved with something that isn't peer review itself, but is certainly thinking a bit more critically about papers and be involved with that community discussion. So with that, I'll wrap up. Thank you very much, and I'll be happy to answer any questions you have in the discussion time or speak more broadly about this. And thanks again to Highwire for inviting me to speak.
Thanks very much, Helen. We'll now go to the panel. So if everyone would on the panel would turn on their cameras. Thank you. And the first question to the panelists is a pretty general one, but I will call on you individually to answer it, and I'll start off with Helen and then go back through the reverse order of the speakers. You've all talked about how the particular service that you've just described talks and connects with preprints. How do you expect that it would connect intellectually, procedurally, technically with the process of journal peer review instead? Helen?
Yeah, well, as I said, we view Prelights quite differently from peer review because it's more about the community discussion, I suppose. And if you read a Prelights post, they're not critical in the way that peer review might be. They're not looking at the methods or the stats, that researchers are using. I think they tend to fit more into the wider context of research, and we actually have done a meta-analysis looking at the type of questions that Prelighters ask, and they do tend to be more contextual, looking at how this research fits into the published landscape already. So I personally view Prelights as being quite different from that journal peer review process, but I do think the more we have these initiatives that think about community discussion around preprints, that can only be a positive thing in terms of improving manuscripts before they do come to that formal peer review when it comes to journal submission.
All right, thank you. So if I were going to write a commentary on a paper that had been pre-lighted, I'd probably want to take a look at the pre-light then for the commentary. Irache?
Yes, so that's a very good question. For the crowd preprint review trial, we don't have necessarily partnership with journals. Essentially, our main goal was to say, can we do review in this model for preprints and encourage participation on the reviewer side. Having said that, there's been some questions from authors, or we had authors interested in social media, saying, actually, can I just get this review and include it in my submission to the journals? Essentially, we provide the reviews for the benefit of the authors and to help reviewers develop their skills. So we very much encourage authors who submit to journals to include it if they wish. If after the trial we were to think about expanding the model, perhaps longer term, larger scale, I think that's something that we will probably consider in terms of incentivizing participation on the author side, of saying, can we find some partnerships with journals so that there is more portability for these reviews when you submit to the journal itself.
All right, thank you. Rich?
Yeah, this is a good question, and I have kind of a scared answer. Is that I don't know that archivists should be integrated into a publishing workflow. I think it's useful to be able to type in keywords related to your research and say, which papers are people talking about right now. But there are a lot of things that are confounded with number of tweets. Sometimes it's people are really excited about something. A lot of times it's people are really mad about something. But it could also just be the senior author has a lot of Twitter followers. And I don't know that that's something journals necessarily want to optimize for. It, I think, could be useful as a demonstration of how much easier it's gotten to pull data from places you're interested in and combine them together. The Crossref Twitter and social media interaction stuff is all publicly available. The bioRxiv stuff is all publicly available. And a script that matches up the entries from both of those things is what generates the archivist homepage. So if there are things that you're interested in or excited about from these services, there's probably ways to use DOIs or publication IDs or titles if you're really unlucky to answer questions that you're most interested in.
So I can sort of imagine a future version of Archivist. If I were a journal associate editor, I'd configure Archivist to focus on my particular specialization in a journal, but somehow have you bring in other kinds of metrics than tweets, things that I might use to trigger, I should be looking into this paper, or even more likely this author, for future papers. Thanks.
Yeah, definitely.
Josh?
Yeah, so I think, our citations can be used as kind of like an early form of peer review, oftentimes going beyond peer review. If someone is replicating experiments or looking at the same phenomenon and they're writing an entire paper and citing it, arguably that could be more. And so that's one potential. Another is that it could be a signal for, soliciting and these authors to submit to the journal. And then the third kind of potential use, I would say, with more qualitative and citation context, would be sourcing peer reviewers. So looking at this reference check, not to check the reliability, but to see who they've referenced and who has also cited that. And we already see that starting to happen actually at journals, and it's not what the tool is designed for. So you can tell there's a strong need for this.
Good. Let me mention to attendees that if you have questions you want to pose to the panel, go ahead and open the Q&A by clicking the button probably at the bottom of your screen, and go ahead and type in the question. We'll pick it up. Let's see, Anita?
So in terms of the the usability of SiteScore inside of journal publishing pipelines, I'm happy to report that at several publishers it's already a part of the pipeline, so that's great. But in terms of how the reviews that are posted on the preprints are actually being used, I've heard varying things, like people have been looking at them from the journals, but often people still don't understand that they are there. So adding a little bit more, and they're present in Society, they're present in the hypothesis group. They do get a lot of views from the, at least from what we can tell are the readers of the preprints, but we haven't seen a lot of those yet incorporated into any kind of a formal review process. We would love to talk to anyone that would like to kind of include those more formally. I think they're valuable bits of information. And one of the things that, we did this in order to try and improve the manuscripts. And I think without, putting them into the kind of more formal processes, I don't think that they will actually do what they're intended to do, which is to really improve those manuscripts. So I very much look forward to engagement with anyone who wants to engage with us for the goal of improving the manuscripts by using automated review.
All right, thank you. Tim?
Yeah, so thinking about sort of journal peer review is sort of a bit like three buckets, in that there's, the three tasks the journal's trying to do. There's the assessment of fit and sort of importance to decide whether the article really should be published in that journal. There's the assessment of robustness, where they try and work out whether the results are supported by the data sets. And then there's compliance checks that the journal often takes on behalf of other organizations. So a plagiarism check is something that they do internally for them. A lot of medical journals check for conflicts of interest, they check for pre-registration, and so on, and ethics statements. And so these are checks that are taking place within the editorial office to make sure that the policies of the journal, of the field, of the funder, of a whole, or of science in general, are being followed. And it's that sort of latter one that DataSphere feeds into. If the journal has a data sharing policy, then if we have already checked that the data are open and the authors can demonstrate that, then the article just flies through that process. Otherwise, the journal would have to go ahead and check that the data have been made available.
Thanks. And John.
Well, obviously, I have a slightly, different view. Not a different view, but I mean, from where I'm coming from is a great deal of pleasure in seeing the emergence of the kinds of services that we have heard about today. A couple of years ago, Richard Sever and Mike Eisen and I published a paper in PLOS Biology, which the main thrust of which was to point out that preprints offered universal access to research results, which at the time, was a major topic of conversation around Plan S and similar initiatives. But a sort of secondary paragraph in that paper said that if preprints became more mainstream, then that would facilitate the emergence of experiments in peer review, the modification of how peer review was done by journals. And that was only a couple of years ago, and already we see the kinds of initiatives that have been talked about and others that there weren't time to fit in today. So I think lots of things are changing. The next task, it seems to me, as has already been said, is to try to be better at integrating the results of the analysis and evaluation of the preprint version of a manuscript and bringing that into the consideration that a journal brings to a submitted manuscript. Very early experiments were done by PLOS in trying to incorporate comments from bioRxiv. It was not a success because, well, for a whole variety of reasons. Commenting is not a very well-recognized part of how scientists engage with any kind of publication. But we shouldn't be discouraged by that. I think there are lots of possibilities, and all of the services that have been talked about today, one can imagine being brought to bear on a manuscript that eventually arrives at a journal with all of that associated information that is helpful to the journal in making peer review faster, more efficient, more insightful, and ultimately better for the community as a whole.
Yeah, I think that was one of my points, is that preprints becomes a lab, if you will, for the journal community to see these experiments happening with authors and see just how well or not they're working. So we have a question from one of the attendees. The question is the principles of the FAIR process in preprint services. Would somebody like to talk about that? You can raise your hand or just speak up.
Well, I will raise my hand to confess that I am not as familiar with FAIR principles as I should be. And so I'm passing on answering that question in favor of the more expert views on the panel.
Anyone? Tim.
If, especially as applied to data, FAIR is very much about ensuring, well, component of FAIR is making sure that there is that link between the article and the data set, and between and perhaps even the section of the article and that particular data set. So it's findable from the context of the article. You can go to the data set. and that the article itself acts as a sort of a metadata point for the data set. This is the context of the experiment that this data set was collected in. So there's a lot that could be contributed to Findable, for sure.
All right, thanks. So with that, since we're just about out of time, let me return the webinar to Allison.
Okay, well, that covers it. Thank you so much for your time today. We appreciate all of the important contributions from John, from our presenters, and from everyone who asked questions. We thank you. We hope you stay healthy and well, and we hope to see you at our next event, which is that OA Week event that I mentioned, and you'll receive a mail about that. So please keep an eye on your inbox and make sure to share your feedback in the survey that we send. Once again, thank you and goodbye for now.